Sister Angela, how are you? I decided to make a video for you to make things easy for you to understand about muscle relaxation. Now remember, it first starts with muscle contraction and contraction. If you understand muscle contraction, just know that the opposite of muscle contraction is muscle relaxation. So you should also understand the key players in each of the two events. So here we have muscle contraction. So under muscle contraction we have key players. Key players we have the first key player is electrical signals, action potentials. Then the second key player we have the calcium ions. Then the third key player we have acetylcholine, a neurotransmitter. Then the fourth key player we have nicotinic receptors. Then the last one we have the sodium ions. So these are the key players in muscle contraction. And then under muscle relaxation, what are the key players? So the key players we have inhibitory neurotransmitters. So inhibitory neurotransmitters we have GABA, and also we have glycine. We have glycine. Then the other key player we have an enzyme, acetylcholinesterase. Then the third key player we have potassium ions. Then the last key player we have ATP for active transport. I'll explain when it comes to active transport. So these are the key players. So once you understand muscle contraction and the key players involved, remember it is just the opposite. Opposite is relaxation. So let me quickly take you through contraction. Just quickly. So number one, we need an action potential. So an action potential has to come down through the axon straight to the axon terminal. So once it reaches the axon terminal, it will cause the voltage gated calcium ion channels to open. So once these gates open right into the synaptic space. So once they open, they cause the synaptic vesicles. These vesicles, they contain the neurotransmitter acetylcholine. So these vesicles, they'll be forced to move towards the active zone. So what is the active zone? The active zone is just the presynaptic membrane. So once it reaches there, it releases the acetylcholine by exocytosis. Exocytosis is just this one is just the fusion of the vesicles with the membrane to release its contents. Exocytosis. So number 4, what happens? The fourth stage, this acetyl, write it like this shortcut. So this acetyl would diffuse, it would diffuse into the what we call the synaptic space or the cleft. So once it goes into that space, it goes to bind. Remember this one here, nicotine receptors, they are also key players. So the acetylcholine will bind with the nicotinic receptors. So after binding, what will happen after this bind? So this binding causes the voltage gated sodium ions channel to open. So once sodium now is allowed to enter, it will cause, number 7, it will cause now depolarization. So after depolarization, the muscle will reach threshold, and an action potential will be achieved, meaning the muscle will contract. So that's the summary I can give you on muscle contraction. So starting from action potential, it goes down through the axon to the axon terminal, which causes the gates for the calcium ions to open. So these ions, they will force these synaptic vessels in the synaptic space, in the synaptic membrane. It will move towards the synaptic membrane and fuse with the membrane, release the neurotransmitters inside through the process called exocytosis. So what will this neurotransmitter do? It will bind with the nicotinic receptor. Now these nicotinic receptors are found on the neural junction plate on the muscle fiber. So once that happens, calcium is allowed now to enter the muscle. So once sodium enters the muscle, it causes depolarization. So this process here, starting number 6 and number 7, this is where you draw that table talking about the action potential, like that here, where it says sodium influx as it enters into the muscle, like that. This is the muscle. So number 6 and number 7 is talking about this table action potential. So you only draw this table if the question is talking about action potential. But if it's talking about muscle contraction, you just simply explain this the way I've explained it, leading to the muscle contracting. So this is the easiest summary I can give you on muscle contraction. So once you understand this one, it will be easy now to take you through muscle relaxation. So under muscle relaxation, remember it's the opposite of muscle contraction. Remember the key players of muscle relaxation, they are inhibitory neurotransmitters. So what happens? Number one, what happens during muscle relaxation? In muscle contraction, there's an action potential that causes calcium ions to enter the neuron itself. Now, the opposite is we don't want any action potential to be present. So the motor neuron, remember, motor neurons are the ones that take impulses from the brain to effect organs like muscles. So we need this muscle neuron, motor neuron, to stop firing, signals, electric signals, which is action potentials, to the muscle, fibers. So that's the first step. That's what we need. So these motor neurons, once they stop firing action potentials along the axon, remember, action potentials pass through the axon of a neuron. So once this happens, we'll go to step number 2. What will happen if there's no action potential coming along the axon? It means the voltage gated calcium ion channels will not open. What does that mean? If these ions are not allowed inside, it means the synaptic vesicles, remember we said these vesicles contain acetyl. They contain acetylcholine, a neurotransmitter, like that. So without calcium, these vesicles, they will not be allowed to fuse with the synaptic membrane of this neuron. So this is the process. So without these vesicles going to the membrane to release acetylcholine, we go to the 4th place. So the 4th point is saying without acetylcholine, there will be no binding. There will be no binding of acetylcholine to the nicotinic receptors. Remember, in muscle contraction, we said when there's binding of acetylcholine to nicotinic receptors, what is released? Sodium is released. Sodium is released into the muscle fiber. So in this case, this gets the voltage gated sodium ion channels will close. They'll close slowly. Allowing potassium gates to open. So at this point, we are talking about repolarization. So if you remember that table for action potential like that, this point number 5, where we've reached, it's actually here, where there's repolarization here, where there's potassium efflux and sodium gates closes. They close. So remember, this is the resting membrane potential. So the resting membrane potential depends on potassium leaving the cell, making the cell more negative inside, making potassium more negative inside. That's why here the millivolts, they are negative. So repolarization is making the cell become more negative because potassium is leaving the cell. We are at this point. Remember, this part, this is where muscle begins to relax. So we are going to point number 6. So what will happen in point number 6? We are still going back to calcium. What is calcium doing? What calcium does, it forces these synaptic vessels to release acetylcholine. But I want you to remember that in muscle contraction, this neurotransmitter, it was already released. So it's still in the synaptic space. The space between the neuron, and the muscle fiber, it's still there. So that's why we said one of the key players in muscle relaxation is an enzyme that breaks down acetylcholine. So we'll talk about that enzyme, which is acetylcholinesterase. So it breaks down acetylcholine into choline and acetic acid, which move back. These two, they move back into the neuron. The presynaptic neuron. I've written presynaptic neuron. So these two, choline, acetic acid, goes back into the presynaptic neuron, back into the vesicles. The vesicles are waiting to be released in muscle contraction. So this enzyme is also a key player. Then point number 7, let's talk about the calcium. Where is calcium during muscle relaxation? In muscle contraction, calcium was helping to release acetylcholine. But at this point, where is it? So this this calcium, it's stored in the sarcoplasmic reticulum. So I'm going to draw a cell here. I'll draw a cell. Let's pretend this is the sarcoplasmic reticulum, the one that stores calcium ions. Then this space, this is the sarcoplasma or the cytoplasm of a muscle fiber. So this is where we have the myofibrils. The myofibrils, if you remember the myofibrils, we have actin and myosin, if you remember these two filaments. So calcium comes from its storage point, which is the sarcoplasmic reticulum. It goes into the sarcoplasma to bind inside you, to mix with actin and myosin. So during muscle relaxation, this is what happens to calcium. Because there's no action potential that is allowing calcium to be released into the into this neuron, what will happen is this calcium concentration in the sarcoplasma will reduce. In here, in this part, calcium will reduce. So once it reduces, we have the ninth point. What happens is calcium will move back into the sarcoplasmic reticulum via active transport with the help of ATP. ATP is also a key player. So this calcium, it will go back where it is stored. Now, what was calcium doing in the sarcoplasmum? Let's talk about what calcium was doing there. Okay, remember we have these actin filaments and myosin filamentine. So point number 10, as calcium reduces, when calcium reduces, it releases troponin. It releases troponin. Once troponin is released, troponin excuse me. Okay, so once it releases troponin or unbinds from troponin, it causes troponin to allow tropomyosin to cover the active binding site for myosin on the actin filament. So this is what happens. So remember, calcium holds on to tropomyosin. Once it holds on to tropomyosin, it forces this tropomyosin. Calcium holds on to troponin, sorry. Once it holds on to troponin, it causes tropomyosin to uncover the actin, the active binding site, that is in muscle contraction. So in muscle relaxation, it's the opposite. Calcium leaves, then it releases troponin. So once it releases troponin, troponin now allows tropomyosin to cover the active binding site. So what happens now after this binding site is covered by tropomyosin? So it will force okay, so it will force the head of the myosin filament to detach. It will detach from the active binding site on the actin filament. So after this detachment happens, an ATP molecule is added to the head of the myosin filament. So what else happens? Cross bridges. You know, cross bridges will not occur. So cross bridges, when cross bridges occur, they allow the muscle to contract. But without cross bridges occurring, the muscle will not be able to contract. So in muscle contraction, cross bridges occur. Cross bridges, this is where now the actin filament slides over the myosin filament. This is what happens here. So in muscle relaxation, because tropomyosin is covering the active binding site, so these two, actin and myosin, will not be able to bind. So a cross bridge will not occur. So what happens as a result? So as a result of these events, the muscle relaxes and your essay is done, and your essay is completely done. So take you back again to the key players in muscle relaxation. Inhibitory neurotransmitters, we spoke about them. And then there's this enzyme, we also mentioned what the enzyme is doing. Then what about this potassium? Here we are saying potassium ions effluxing, as in leaving the cell, very important. Then ATP, remember ATP was helping here. ATP was helping in transport, active transport, moving calcium ions from the sarcoplasma back into the sarcoplasmic reticulum. So this is also another key player in muscle relaxation. So once you understand muscle contraction, you'll be able to understand muscle relaxation. So the way the questions are asked, there are three things, the way the question may be asked. So the question can be asked about muscle contraction. So muscle contraction, I took you through it earlier. Then there's muscle relaxation. Then the question can also be on action potential. Here, this is muscle excitation, which leads to muscle contraction. So you have to be careful how the question is asked. Are they talking about muscle contraction, which is muscle excitation, where you talk about the neurotransmitters on the neuromuscular junction, or they are talking about muscle relaxation, or is it action potential? So I hope this is a bit easy for you to understand.